Alright, welcome back. So we're going to work through our next type of formed element, which is the leukocyte or white blood cell. Leukocytes, again, two main groups on these ones. You have the granulocytes, which have membrane-bound visible cytoplasmic granules. These are neutrophils, eosinophils, and basophils. Granulocytes do not have these granules. A meaning no granulocytes, no granule cells. These do not have these granules. These are going to be the lymphocytes and monocytes. So one of the ways to me to kind of easily remember this is if you kind of go Phil eats grains. If Phil eats grains, he has grains in him. So all the granulocytes end in Phil. So Phil eats grains. So Phil has grains. The other way to remember this, no grains in sight is the other one here. So if it ends in sites, there's no granules in a cell name that ends in a site. So that's an easy way to remember granulocytes versus agranulocytes. So this can kind of showing you all the different ones that are on there. What we're going to do is work through these one by one. There's, again, per microliter, 5 to 10, 4 to 11,000, again, depending on which textbook you look at, but somewhere around there. And again, leukocytes are really here to be some type of, it's part of our immune system, part of our immunity. They are going to be these cells that are doing different things to get rid of bacteria, viruses, parasites, other things like that. Cancerous cells is really why we have these in place. And again, that is their main function. When we talk about the different functions of the circulatory system, leukocytes are really doing that protective function. Depending on what type of leukocyte we're talking about, its lifespan can be very short to way longer. Certain memory cells can last years or close to a lifetime. So again, depending on what we're talking about, there's going to be some differences on these. So what I want to do is walk through each of these types and talk about what it does. Again, I will try to point out to me what is the key thing on these ones. So overall, you're looking at somewhere about 50 to 60% of white blood cells are going to be neutrophils. So easily the most common type, definitely something I want you to be aware of. These ones, the granules are there, but they're not super obvious. So of all the granulocytes, probably the one that has the least obvious granules. So what makes this one probably the most distinct is its multi-lobe nucleus. 3 to 5 lobes on the nucleus. Sometimes they also call these polymorphonuclear leukocytes. So polymorpho, many-shaped nucleus leukocytes. Not as big a deal. The duration of development takes a few days to about a week. They can last a few days in circulation, but again, the more important thing here is their main job is to phagocytize bacteria. So these are going around. If they run into a bacteria, they will engulf it and break it down. So these are phagocytes of bacteria. So to me, kind of a picky eater. If they take something in, they're going to take this, fuse it with a lysosome, break it down. The other thing they're going to do, which we'll talk about with the immune system, is kind of show a little bit of what they ate on the surface of the cell as a way to trigger the rest of the immune system. Again, we'll get into this more with the immunity when we take a look at those videos. Eosinophils are really the next to last most common, so these are the second most rare. It's about 1 to 2% of white blood cells. What makes these ones incredibly obvious is they have very distinct granules that take in the eosin or reddish pink stain really, really well. So you're going to see a lot of these reddish pink granules in the cytoplasm. They tend to have a bilobe nucleus, either like the one shown here, like a headphones, or a barbell nucleus. So barbell means two sides here with a little connector in the center. Again, about a week to develop, lasts a little over a week to 2 weeks. Main job in these ones is they are involved in getting rid of eukaryotic pathogens. So things like parasitic worms are kind of when you'll see these kick up the most common if you have a worm infection. Again, not something you probably ever want to hear somebody say. By the way, you have worms. Also involved after an antibody reaction. And we can see some inactivation of some inflammatory chemical responses of allergies. A lot of times after you have an allergic response, you'll see the eosinophils come up in number. least common type of leukocyte is the basophil. It does have a lobe nucleus. It's very difficult to see it because it has these very large, distinct purple granules that you can't really see the nucleus underneath in there. Again, about a week to develop, can last hours to days. Again, not so worried about the development and the lifespan of them so much as I am about what they're doing. So these ones are a blood cell that is going to trigger an inflammatory response. So they release things like histamine and heparin. So anticoagulant is heparin, lets more blood flow to get to the particular area. These are active. Histamines are going to trigger an inflammatory response. And again, we'll talk about what inflammation does in the immune system when we get to the immune system. But these are going to be these cells that can kind of trigger this. I always kind of say they're like a flare cell. They send a shot into the air saying something's going on here. We need more immune cells to get to this area. So basophils, like I said, that histamine release causes more inflammatory response, more permeability, more blood flow to the area. Heparin stops it from clotting in that area, so more immune cells can get to this area. If you've ever taken an antihistamine, you know it's going to deal with, like, if you have an infection in the nose, some type of cold, for example, it will open you up a little bit more with those antihistamines, histamines, more permeability, more flow of liquids to that area, swelling, things like that in that area. The 2A granulocytes are the second most common and the third most common types of leukocytes. So lymphocytes are T cells and B cells. These are about 25 to 35% of white blood cells, depending, again, on which textbook you look at a little bit. Tend to be the smallest cell and either have a nucleus that takes up about half to somewhere around 70 to 90% of the whole cell. Either very circular or kind of half the cell taken up here. These take usually a week to 10 days to develop, and these are the ones that are capable of doing immune memory. Because of that, some of these can last 2 years in terms of how long they live. This is your specific immune response. So like if you get a vaccine, so COVID vaccine, for example, if you're getting that, it is triggering these T cells and B cells to make antibodies, T cells to have a direct immune attack on these infected cells. But these are part of that specific immune response. Again, we will talk about this in a lot more detail as we get to the immune system, but this is the job of what these lymphocytes are. And finally, our last type of leukocyte, 5 to 15% is what I would say is probably a pretty safe range on these ones. They tend to be the largest of the leukocytes, usually have a U-shaped nucleus that's decently large. They can live a few months. These ones are very similar to their job to what neutrophils are doing. These are phagocytes. Again, these ones though, if they get on the tissues, we call them macrophages. They are going to be engulfing a lot more things than just bacteria. Damaged cells, cellular debris, cancerous cells, things like that. These monocytes are a little bit more involved in what they eat. So one of the ways we can kind of sometimes tell what's going on there, if somebody has an infection and we know something's going on, but we're not really sure what, a lot of times what they'll do is what we call a Diff Count or a differential count. What they're doing on these ones is they're taking a blood sample and giving you the relative percentages of these leukocytes. Like I said, normally 5 to 10,000, 4 to 11,000, somewhere around there per microliter. If we see higher numbers overall, that's saying an infection. But what we can also look at is the relative percentages of these ones. So normally, like we said, 50 to 70, 20 to 40, 2 to 8, 1 to 4, less than 1%. If all of a sudden we're seeing eosinophils are way higher than normal, and we know that these are dealing with parasitic worms, for example, that might say if you have 8% eosinophils, you have some type of eukaryotic pathogen. Maybe you have a parasitic worm infection, where if your neutrophils are higher than normal, they might say, okay, those are phagocytes of bacteria, you are having a bacterial infection. So this is part of that diagnostics in terms of using these as information. If you can kind of get these relative amounts, and if something is out of its normal range, that can tell you a little something. So like I said, neutrophilia, that's too many, more than normal neutrophils, that could be a bacterial infection. Lower than normal, that may mean you have an anemia or some type of reaction. Higher number of lymphocytes might be a viral infection, a leukemia. Too low one might be something like HIV. So that lymphocytopenia. Eosinophil change, like I said, allergic reaction, parasitic worm infections, monocyte changes. High increases would be a chronic inflammatory thing, something like tuberculosis. Lower than normal may mean that you're being treated with some type of steroid response to tone down your immune system. And again, basophil ones here, generally it is some type of overproduction of bone marrow, some type of disorder. Sometimes you'll see these up in response to acute allergic reactions or stress reactions. You'll see a decrease in these. If these leukocytes need to leave the bloodstream, it is a kind of multi-step process for this to happen, especially when we start talking about monocytes becoming macrophages. I always kind of say this is a lot like getting off the highway. If you think about the bloodstream as like this big highway with all these blood cells moving through it, a lot of times if you want to get off the highway, the first thing you need to do is get into that right lane and be able to slow down. That's margination. These blood cells tend to get to the side of the blood vessel and then start using some adhesion molecules to slow down as they roll along the wall of the blood vessel. The next thing you need to do on the highway if you're going to get off is take your exit. That's diapedesis. These leukocytes slow down and now take their exit between some of the spaces in between the cells of these endothelial cells and make their way out of the bloodstream. After that, you may want to follow directions to wherever you're going. That is that chemotaxis. A lot of times with inflammation or an immune response, you have inflammatory chemicals that are going to be in the highest concentration where the actual infection is. The release of these chemicals, these blood cells can actually follow the levels of these ones and home in on the source of those. That's that chemotaxis, which will allow these cells to actually get to the area where the infection is. Once they get there, they can phagocytize this, get rid of these, help clean up that area, and get us back to a normal state. Normally, like I said, leukocytes should be about that 4 to 11,000. Anything beneath that we call leukopenia. Too much of this is leukocytosis. More than normal doesn't necessarily mean an abnormal disease. Leukocytosis can be a normal response to an infection. If we get sustained leukocytosis, that can sometimes mean that you're having some type, especially if it's of abnormal types or undifferentiated types of leukocytes, that can be a leukemia, some type of overproduction of these leukocytes in the blood. Leukopenia, a lot of times, if you're getting treatments for cancer, like chemotherapy, for example, is going to be a good example or a good reason why you might see lower than normal white blood cell counts. The main reason is a lot of these dividing cells, these stem cells, are what are affected by this, and chemotherapy tends to get rid of these fast-growing levels here, and it's those precursor cells that are fastly dividing that are affected by this, and again, can tone down those number of leukocytes. So chemotherapy a lot of times make you more prone to infection. Other things that are dividing very fast, like hair, epithelial cells in your intestine, that's part of the reason you get issues with digestion with chemotherapy. You lose your hair with chemotherapy. It's, again, part of this. The other thing a lot of times you'll see is if it is a leukemia, a lot of times if you're overproducing a particular type of blood cell, you tend to underproduce other things. So a lot of times if you have a leukemia, you might be having lower than normal, you might have anemia happening, you might have clotting issues happening, that little spidery bruising called petechia happening with these leukemias. And leukemia, again, is that overproduction of certain types of white blood cells. Again, different types of leukemia depending on what precursor cell is being overproduced. A lot of times these are non-functional white blood cells, so even though you're overproducing a white blood cell, your immune system is actually impaired. And again, a lot of times it leads to a lack of red blood cells and anemia, lack of platelets, which leads to that bruising. So again, different types depending on which one there is. So ALS, acute myeloid AMS, acute myeloid AML, acute myeloid leukemia is a type. Some of them very treatable, some of them are not. Really depends on the type of leukemia. So what we're going to do on the last one of the blood is we're going to take a look at the platelets and the process of blood clotting. That'll be the last thing we do on the blood. We will then move on to the heart and go from there. So I will see you next time.